All right, so I'm going to be talking about the laboratories done in our biology lab in your at-home online course. So that's just it. There's not online labs in the sense that you're not just doing labs online. Well, the course is an online course, and you're watching lectures like this one at home, and then doing the other assignments online through the Blackboard platform. The laboratories are primarily hands-on, so you have to have your laboratory kit, which I showed in some other little videos. It has all the material, well, not all, but that's what I'm going to talk about right now. It has most all the materials you need to do the labs, but there are a couple things that you're going to have to buy on your own, and there may be things you don't have to buy because they're already in your house. But there's a couple labs where you're going to need just some sugar. So if you don't have regular just sugar, that's what you need. In our labs in person, we just use the same sugar. We can buy laboratory grade sucrose and pay hundreds of dollars for it, which has slightly better purity than this. Or we could just use this and have essentially the same result in the lab. So you're using the exact same thing at home. It's safe. It's just kitchen consumable grade. You will need for one of the labs on enzymes hydrogen peroxide, which you may already have, but it's very inexpensive. It's only $1 or $2 for a bottle like this. You'll also need some isopropyl alcohol for a lab, and I don't have here like a nail polish remover or something like that, which I'll give you more details on with that particular lab, which is the chromatography lab that you're going to do. So right now, the main thing is I'm going to give you a real quick overview of all the labs that you're doing, just to be very clear on what's going on with each of them, okay? Because for all of them, you're going to have some kind of worksheet to submit. For most of them, there is some video or photographic proof or evidence of your work that also has to be submitted along with that, right? And other times it's just the worksheet. There are a few times where the laboratory is very complicated and it's not something that everyone could just do at home. There's no equipment I can provide you that doesn't cost thousands of dollars. So there are labs where I'm going to be providing you with data, and then you're going to do the analysis and the presentation and all sorts of other research on it outside, but not a physical experiment. But most of them will be physical experiments. So that's what I'm going to talk about here. The first one is on microscopy, because we can't get you really microscopes at home. This is one that's going to be over the parts of the microscope, how the microscope functions, how the parts function, and several different calculations on magnification and resolution and field of view. And so you'll be looking at that and some additional video. So that one is kind of like the first one. It's very straightforward and is sort of an online one, just an overview of a microscope. But you're not going to be really using it at home because you don't have one. So we're not going to be essentially doing anything more with that. Pipetting, you are going to be physically using a lot of things in your lab kit. So you'll be needing the little electronic balance. You'll be needing the weigh boat. You'll be needing just some water. You'll be needing a bunch of pipettes. And you're both going to calibrate pipettes as one part of a hands-on lab. And then you're going to secondly then perform data collection, where you're going to pipette liquid and then weigh it. And you can do that a bunch of times with different volumes. And then you're going to do a bunch of calculations after that with your data and submit it. So if you don't have the data, you can't do the calculations. I'm not providing the data for you. You have to get your own data to do it. So serial dilutions and pipetting, that's one that you have to physically do a lot of work with yourself. Serial dilutions, another one. For this one, you're going to need food coloring of your own. So you can see here, these are just tubes that are provided in your kit. You'll need some water, you need some food coloring, and I show you in the specific serial dilution explanation video how to make this. You're going to be making one that looks like this, and a second one, I'm not showing you what it looks like, and you have to submit photographs of both of those. So that's in the serial dilution lab. Serial dilutions like this are sometimes used to create a standard curve, and that standard curve is then where we measure these in a spectrophotometer, essentially how much light is absorbed or how much light can pass through them. Since you don't have a spectrophotometer at home, and I've tried different ways to come up with a device where you could use your phone or an app or something to actually then measure light going through the solution, I'm just going to be providing you with data from this. So essentially, from something that's been created like that, you're going to then make a graph. So I'm going to just provide you with different data that you're going to use to make a standard curve and label it, and the instructions are with that particular lab. So that one is one that's just data that has been provided to you. You're not going to generate the data for the standard curve lab. Molarity is one that you're also going to do yourself. There's a worksheet with a whole bunch of calculations, but in addition to that, you're going to then be using the sugar and your little weigh boat and some of the little tubes that are provided in here. And so one of the things you're going to have to do is, after performing some of the calculations, you're going to have to weigh out sugar, you're going to have to measure out a specific volume of water, and you're going to have to photograph it as a result. So I will be able to check your problem, and if you say you're supposed to weigh out 35 milliliters and you take a picture of this and it only has 25 milliliters, you didn't obviously just measure the water correctly. And I kind of know what these things should look like in terms of the volume. So if you're weighing out 0.2 grams and I see this whole thing looks like it's full of sugar, I know it's wrong. You've done something wrong. So I'll be able to comment back to you and say, that doesn't look quite right. Can you check this or redo it? Same thing. If you're having questions while you're doing some of the work, you'll submit to me and say, this is what it looks like. I don't think that's right. And I'll be able to comment back to you to say, you're correct. It doesn't look right. I think this is what you should do. So you'll be able to submit both the actual results of work, and you'll be able to submit questions you have of work that you may be having trouble with as well. So you'll just send that to me directly through the class email. The osmosis lab, that's one where you're also going to do something physical with, and it's an extension of the molarity lab in a sense, and that you're going to then have to make up several solutions. So you'll be measuring out liquids, you'll be weighing out sugar, you'll be creating different molar concentrations of solution, you'll be putting them in dialysis tubing. This little, you'll just see a little flat piece of clear tubing in here, but you're going to have to make a little bag like this and put your solution inside it, and then you're going to weigh that little bag. Then you're going to put them in containers with water, and then something's going to happen. They're either going to lose some of the water content in here, or they're going to gain water, and it's going to be somewhat dependent upon the amount of sugar that's in each of your little containers. So it's going to be osmosis or diffusion of water, and you're going to then measure that actually and collect data on it with your balance. You can measure how much has moved in or out, and then report those results as part of a worksheet. So that's another lab that you're going to have to do. Protein unknown, this is another data one. So this is one where it starts to get a little more complex, and at the end I'm going to kind of link a bunch of these together to show you how the labs link to one another, one link to another, and so forth. Here you're going to have to create a standard curve, so it kind of comes back up to here. So the same exact thing that's done here has to be done again, but with new data. So I'm going to provide you totally different data and information. You have to make a new graph. This first one up here is just make the graph, label it properly, send it in. I tell you whether it's done correctly or not. You can redo it, or you're good. Here you're going to make another graph, and then there's going to be a series of problems. You're going to have to use that graph to solve for unknowns. All right, so you're going to go a step further than that with this protein unknown. It'll be same thing. I provide you stuff that other students collected in the lab themselves, and that's what I'm going to give you, but then you're going to do the analysis and then the calculations and all your stuff afterward. Catalase is an experiment that you're going to actually do. You're going to need a potato as well for this one. You're going to have to be blending up the potato with water in that lab. Again, I have specific explanations of every individual lab. This is just the overview. You'll need the hydrogen peroxide. You'll be using one of your tubes in here that's graduated. You'll be using the ruler and forceps and a whole bunch of little things that are in the kit. And you're essentially going to then create a bunch of dilutions of your enzyme extract, and then you're going to be testing them for a reaction. So hydrogen peroxide and the enzyme will react to form water and oxygen, and the oxygen bubbles will cause little discs, which are in one of these things here. There's these tiny little discs, which you can't probably see, but you can see something moving inside here. They're filter paper that's going to absorb the enzyme, and then you're going to measure them and time them, physically time them rising up. So you're going to collect data yourself for this that you're going to use to make a graph and do calculations and answer questions. This one is a big, more involved report that's worth more. Okay, so all the individual labs are these 20-point things that you're going to turn in, but this one's going to be worth 40. It's going to be double because there's a lot more that you're going to do. With the tyrosinase one, first I'm going to give you data to make another standard curve. So you're essentially going to be making 3 standard curves with different data sets. One of them will be for a specific dye, the other will be a Bradford reaction with protein, and then this one will be dopachrome, which is a pigment molecule. So there'll be data, you're going to create a graph, the graphs are going to look similar to one another, but they're going to be using different data with different molecules. And they're going to be used in different ways. Just the graph plus one unknown calculation, or there's like actually 3 or 4 different ones to do, but just the same thing over and over again. And then a tyrosinase one where I'm going to give you data first for the dopachrome. Then I'm going to give you another set of data that is part of an experiment where pH was done, pH was altered, and then an experiment was performed over and over again. And you're going to get this data. You're going to have to do a whole bunch of calculations. It's more complicated than I'm going to explain right now. So you have to wait for the other lab. But you are going to still do something physically for part of this, and that's the pH testing. So in this experiment, it's looking at how the enzyme works at different pHs. So one of the things that you're actually going to do is then test the pH of a variety of different solutions. So have a bunch of pH testers inside here, and a way of reading them and get the results. So you're going to have to find different solutions, just household solutions that you're going to have, that you're going to then measure the pH and then report that on it so you can actually see some of the differences in pH. But then there's a whole bunch of other stuff to do with this calculations and 2 different graphs, so it's a lot more work. Pigment chromatography, fairly simple. This is one you're going to then use the alcohol for, water, and then some other solvents like nail polish remover. And you're going to just need a leaf. So it could be spinach, it could be from a tree outside, and you're going to get a paper in the kit. You're going to transfer some of the pigments to the paper. You're going to use different mixtures of solvent solutions to separate them, and you're going to report that back to me with a photograph of your results and the list of what was actually used, in each of them. The mitosis one is a worksheet that consists of photographs that I took of slides in our laboratory. So you're going to see on your sheet exactly the same thing that you would see if you're looking through the microscope, exactly the same. And then you're going to be identifying stages of mitosis. You're going to be counting, you're going to be collecting data, like how many metaphase cells or how many interphase cells. You're going to do some calculations actually with that. It'll help you learn the stages as well. There's a meiosis one that you'll do that's similar to that. And they're kind of linked together. And then there's a bunch of genetics problems. So for these 2 are just kind of worksheets and problems. They're related to specific lab work, but the work that you're going to do is more with the worksheets themselves and at home. So there's nothing physically in the kit that's specifically related to those particular labs. So that's kind of the overview. Like I said, each of these, every single one on here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Those 11 are worth 20 points each, but then this one here is a bigger one that's worth 40 points in the overall lab because it's going to double. There's a lot of extra work. So when you have questions, email me directly and I'll be able to answer them. Show me photographs, take videos, even ones that I'm not requesting, but ones that are, if you have problems, say making this, then show me what you have and then I'll be able to critique that and tell you what you need to do that might be wrong. So if you have every single tube looking exactly the same color, you obviously did something wrong. They shouldn't look like that in that lab. They should look kind of like this with a gradient. So show me your work and then I'll be able to give you the feedback on it.